Hello all, welcome to another session of Rubin Science. In this session, we shall be studying about the prokaryotic cell and the eukaryotic cell. The previous session, we had studied about the brief history about the cells and the differentiation based on the size, shape, and different functions. In this video, we shall be studying with the difference between the prokaryotic cell and the eukaryotic cells. So, as we know, cells are the basic fundamental unit of life, which are mainly categorized into prokaryotic cell and the eukaryotic cell. Here, the key differentiation is based on the structure function. Okay, so here you can see a basic structure of an eukaryotic cell and a simpler structure of a prokaryotic cell. So, to begin with, what does a prokaryote mean? So, to understand this word, let's split this word. Okay, pro means a primitive. Okay, a primitive karyote means nucleus. The organisms whose nucleus is primitive in nature or the basic in nature or the earlier version of the nucleus is called as a prokaryotic cells. The organisms which bear a primitive nucleus, these are called as prokaryotic cells. So if the nucleus is so primitive or the basic version, how does the organism look? So they are simple. They are unicellular organism. They are simple means, yeah, it's a single organism. They are just simple in their structure. They're unicellular. So this whole entire organisms, the entire cell is an individual organism. Okay. So and another thing is they lack nucleus. Nucleus. So this they don't have. Okay. If the nucleus is not there, then how do they transfer the genetic material? That will be studying in further slides. And the membrane-bound organelles are also absent, for example. It's like the mitochondria. These are all are absent in that. Okay, it is not present. The examples are bacteria and archaea. Whereas, what is eukaryote? To understand this, let's split once again. Humans, true. Okay, karyote means once again, it's a nucleus. The organisms who have a definite or a true predominant nucleus is called as eukaryotes. So if they have a definite defined nucleus, then uh, how does the organisms look? They are complex in structure, they are multicellular. See here, a single cell is an individual organism, whereas a multicellular organism, a group of cells together forms an organism. So, okay, for example, you consider a liver like this. So the liver have multiple cells. Here, a group of cells together has formed an organ. I mean the tissue, and group of tissue together forms an organ, and group of organs together forms an organ organ system, and a group of organ system forms an organism. So here, for a single organ or a tissue, there are multiple cells. So these are multicellular organisms. And the main thing, the most important thing is about they have defined nucleus and they have membrane-bound organelles. So what all are absent in the prokaryotes that are all present in eukaryotes. Examples are plants, animals, fungi, and protista. Now, let's start a classification, individual study about their individual characters. Okay, as I said, the prokaryotic cells are simple in structure, they lack nucleus. So, nucleus is not present. Okay. The best examples are like E. coli, Streptococcus in for bacteria, and for archaea it is methanogens and halophils. And for eukaryotes, they have the complex structure and well-defined nucleus. Examples are plants, animals, fungi, and tista. To have an overview glance of an differentiation, nucleus is absent. Okay, it is absent in prokaryote and it is present in eukaryote. Now, I said nucleus is absent. Nucleus is the place where all the genetic materials will be present. Right? So, an alternative form of nucleus, that is a nucleoid. mean, what is the difference between a nucleus and a nucleoid is this is a nucleus where the genetic material is covered or bound or protected with a nuclear membrane. Okay. Whereas a nucleoid is something where a nucleus membrane is not there, but the DNA is present in the cytoplasmic region. Okay, this is a nucleoid. A nucleus, a genetic material without its membrane boundary is called as a nucleoid. Okay, so the DNA is circular. Yeah, here the DNA is circular in nature. Okay, whereas the DNA it is linear. Here linear DNA, and here it is circular DNA. And the membrane bound organelles, right? Membrane bound organelles are absent and they are present. The size is zero point 0.1 to 5 microns, or we can say zero point 0.1 to 10 microns they come up to, and these prokaryotes comes to 10 to 100 micrometers. And the cell division is through binary fission, which means a single cell divides into two identical daughter cells through two equal arms. That is called as binary fission. Whereas this goes with mitosis and meiosis, which is a multiplicative division and the reductional division for sexual and asexual reproduction. So, to begin with a structural differentiation, as I said, these prokaryotes, as their size varies from 0.1 to 10 microns. Okay, these are smaller in size, almost which you can't see directly through your eyes, only you can see through microscope. These are smaller in size in terms of size. In terms of shape, which we have studied in our previous video, that is the one taken with Tokai shape, rod shape, which is a bacilla, and a spiral shape, which is spirulina. And nucleus is absent. So nucleus is replaced by a membrane called another is called as nucleoid. Okay, where the nuclear membrane is absent, which means a genetic material is freely floating in the cytoplasm. Okay. Next, membrane-bounded organelles, they are also absent. Which are all absent membrane-bounded organelles? Mitochondria is not there, endoplasmic reticulum is not there, and Golgi apparatus are not there. And ribosomes, the main function of ribosome is a protein synthesis, okay? Protein, for mainly for protein ribosomes. So here we have 70S ribosomes, okay? And the cytoskeleton. Cyto means cell, skeleton means a structure, what we can say. Generally, the cytoskeleton of prokaryotes are simple, okay, and they are less developed. When we compare to the prokaryotes, they are less. When they compare to eukaryotes, they are less developed. If you say the cytoskeleton is less developed, then what they are made up of? I mean, they have a cell wall as their outer layer. So the cell walls are of mostly the. In prokaryotes, they are rigid cell wall which are made up of peptidoglycan. Peptidoglycan. Peptide means a lipid or fat layer. Glycan means a saccharide layer. Okay. These are made up of peptidoglycan, means a fat and saccharide combination of layer. This is in case of bacteria. In case of archaea, it is made up of pseudopeptidoglycan layer. Pseudo means false. It's a false pseudopeptidoglycan layer. Okay, this was about structural differentiation in prokaryotes, whereas in eukaryotes, it is dead opposites of prokaryotes. Okay, so the sizes are larger, which is ranging from 10 to 100 micrometers or the microns. And the shape, they have different vibrant shapes. Here we can just see a broad spherical, oval, or a spiral shape. Here they are diverse shape. They are more complex in structure. They have irregular in structures like that. And the nucleus is absent in case of prokaryote, whereas it is well defined and predominant in eukaryotes. And they contain a genetic material which is enclosed in a nuclear membrane. And the DNA it is linear. Okay, whereas a DNA in eukaryotes it is circular and it is linear. what things on the circular DNA we'll be studying it in further slides. Next, membrane-bound organelles, which is absent in prokaryote, that is present in eukaryotes, which includes lysosome, Golgi apparatus, endoplasmic reticulum, mitochondria, and everything. And the ribosome in eukaryote it is 70s, whereas it is 80s in eukaryotes. Okay, and cytoskeleton, it is well developed. Okay, they contain. Okay, it is simpler in prokaryotes, right? It is well developed in prokaryotes, which consist of the microtubules, actin filaments, and intermediate filaments. And the cell wall. Okay, the cell wall, they are present only in plant, fungi, and some protista. Okay, so in fungi, it is made up of chitin, and in plant, it is made up of cellulose. Okay, this was about the structural differentiation between prokaryotes and eukaryotes. Next differentiation is genetic material. As I said, both also have genetic material, whereas they are circular in prokaryotes and they are linear in eukaryotes. So DNA is circular and they are located in the nucleoid region, which is not enclosed by a nuclear membrane. So they are freely floating in the cytoplasmic region. Okay, and they have plasmid. Okay, so what is plasmid? They helps in plasmid is generally a circular DNA molecule, okay, which helps in gene carrying. Okay, they also carry the antibiotic resistance gene and everything. So where nowadays we are hearing about AMR, right? That is antimicrobial resistance for that. And the chromosomes, they are typically singular chromosomes, and some prokaryotes have multiple smaller chromosomes also. In case of eukaryote, the DNA is linear and enclosed with a membrane. Okay, it is linear and they are enclosed with a membrane-bound organelle. Okay, that is a nuclear membrane. And plasmid is not present in eukaryotes, but some certain organelles like mitochondria, they may have internal material as a plasmid. So this you can see they have only few nucleus and like they are freely floating in the cytoplasmic regions. Here they are enclosed with a membrane. Next is in terms of reproduction, as these are simple, as eukaryotes are, I mean, as the prokaryotes are simpler in structure, they more asexual type of reproduction, okay, which mainly is through the binary fission. Binary fission is just a single cell. They split into two identical daughter cell. Okay. Here it is exactly an equal halves. Okay. Here central part you can take yes, I mean the nucleoid is divided into two equal daughter. It is an identical daughter cells. So why it is identical? Because you can't see much genetic variation here, as the single parent is involved in the reproduction. Okay, and some variation you can see in terms of horizontal transfer occurs where in form of conjugation, transformation, and transduction. Okay, where conjugation is like and cell, there can sex pillar, where the genetic material is transferred to an other organism, and I mean the other cell, the same species, and there can find genetic variation. Next is in eukaryote. They are both sexual and asexual type of reproduction. Okay, where they go both mitosis and meiosis. Okay, mitosis and multiplicative asexual reproduction, and meiosis is in sexual reproduction, where it is mainly used in gamete production, mainly like sperm and egg. And here you find major of the genetic variation. Why? Because two parents are involved. Two parents are involved. The reproduction where male and female gametes involved to form a new offspring. Okay. Coming to metabolism. Metabolism is nothing but breakdown or the energy production, how ATP is produced. Okay. In prokaryote, energy production is generally primarily through a process called as anaerobic respiration, and even through aerobic respiration through fermentation, where how your idli batter or the wine production that are produced, right? That is the method where energy is produced. And some bacteria also use photosynthesis for the energy production. And enzyme localization, prokaryotes use the enzyme for energy production located on the cell membrane as they lack internal organelle. Whereas in eukaryotes, yeah, enzyme localization, like which occurs in glycolysis, occurs okay in the cytoplasmic layer, and even in mitochondria and mitochondria for aerobic respiration, like that. Whereas energy production in eukaryotes are generally through the cellular respiration, which mainly occurs in the mitochondria, which is an aerobic process. Cell division, uh, yeah, the cell division, as we studied initially, it's a binary fission, which without any spindle fibers. This is important. It's a binary fission without any spindle fibers. Whereas in eukaryote, they have both mitosis and meiosis. Okay, and spindle fibers are there. And what is the function of spindle fibers? They help in chromosomal separation. And the best examples for prokaryotic cells are bacteria. Bacteria are the best examples, which includes E. coli, Salmonella, Streptococcus, whereas in archaea, Thermococcus, Halobacterium, these are all the examples which can live in the shallow lakes, salty lakes, and all. Okay. For eukaryotes, which we commonly know animal, plants, fungi, these are best known examples like tiger, human, fish, birds, plants, trees, flowers, fungi like mushroom molds, aspergillus, and protista like amoeba, parameciums are the best known examples for eukaryotes. Evolutionary difference, like as we know, prokaryotic cells are considered to be the oldest and simplest structure. That's why they are called as a it's a primitive structure, and they appear around 3.5 billion years ago and are typically unicellular, whereas eukaryotes are developed from the prokaryotes, which are living from 2 billion years ago. Okay, and they have some symbiotic relationship. It's like endosymbiotic theory, which states about that. Where the prokaryotic cells are engulfed by an eukaryote and evolved into an organelles like mitochondria and chloroplast. And complexity, as we know, prokaryotes are simple, they are smaller, and often have single function or task. Whereas eukaryotes are more complex, they are multi-organal, multi-structural, they are like a specialized function for specialized organs and allowing for higher order of structures like multicellular organisms. This is a key summary between prokaryote and eukaryotes, where their sizes are from 0.1 to 10 microns, and they're 10 to 100 microns. Nucleus are absent, where nucleoid is present, and here nucleus is present with the nuclear membrane, and membrane-bound organelles are absent, and they are present here. And the DNA is circular here, and it's a linear DNA here. And the ribosomes are 70s, which is quite smaller, and it is a larger subunit with 80s. And the cell division is through binary fission, and it is like mitosis and meiosis here. And the metabolism is diverse, includes anaerobic respiration. Here in eukaryotes, mostly aerobic respiration, which occurs in the mitochondria. And the examples are bacteriarchaea, and the examples for eukaryotic cells are animal, plants, fungi, and protista. Thank you for watching this video. And this session we studied about the difference between a prokaryote and eukaryote. In coming sessions, we shall be studying more about biology. Thank you for watching.